of breeding self-pollinated crops. This chapter will explore the fundamental concepts and techniques used in the breeding of self-pollinated plants, focusing on improving crop varieties. Section 6.1 introduces selection in self-pollinated crops. Selection involves isolating desirable plant types from a population. It requires variation in the population and heritability of these variations to be effective. There are two types of selection: natural and artificial. Natural selection is influenced by environmental factors, while artificial selection is human-directed, such as through progeny tests, which are crucial in modern plant breeding. Selection methods in self-pollinated crops include identifying superior genotypes and multiplying them. Common methods are mass selection, pure line selection, pedigree breeding, backcross breeding, and bulk populations. Mass selection is one of the oldest plant improvement methods. It involves selecting a large number of plants with similar phenotypes and mixing their seeds to create a new variety, focusing on observable traits like plant height and disease resistance. Mass selection is limited by the genetic variability of the original population. It can be positive or negative, aiming to either preserve desirable traits or eliminate undesirable ones, resulting in a more uniform population. This diagram illustrates the diversity of plant phenotypes within a population, highlighting the importance of selecting the best candidates for breeding to improve crop characteristics. Here, we see a uniform group of plants representing the outcome of successful selection processes where desirable traits have been consistently bred into the population. The mass selection procedure spans several years. Initially, phenotypically similar plants are selected for desirable traits. Over subsequent years, these selections undergo yield trials, leading to the release of a new variety by the 7th year. Mass selection has merits such as improving local varieties and purifying existing lines. However, it cannot generate new variability and may not distinguish between hereditary and environmental influences on phenotypes. Pure line selection, proposed by Johansen in 1903, involves selecting seeds from a single homozygous plant. This method ensures genetic uniformity, with any variation being environmental and non-heritable. A pure line is a progeny of a single homozygous self-pollinated plant. All plants in a pure line are genetically similar, with variations due solely to environmental factors, making pure line selection ideal for improving landraces. Genetic variation in pure lines can arise from mechanical mixtures, natural hybridization, or mutation. However, all plants within a pure line share the same genotype, with non-heritable environmental variations. Pure line selection is applied to improve self-pollinated crops, enhancing local varieties, old pure lines, and introduced varieties. This method is crucial for maintaining genetic consistency in crop improvement. The general scheme of pure line selection involves selecting a large number of plants based on phenotype, growing progenies in a plant-to-row basis, and critically evaluating seeds to select the best entries. Further steps in pure line selection include testing bulk seeds in preliminary trials, evaluating progenies in replicated trials, and releasing outstanding varieties. This process ensures the development of superior crop lines. Pure line selection offers advantages such as extreme uniformity, stability, and ease of identification in seed certification. These traits make pure lines attractive to farmers and consumers alike. However, pure line selection has limitations. It does not create new genotypes, has limited adaptability, and requires significant skill and time from breeders. Improvement is capped once the best genotype is isolated. Pedigree selection involves selecting individual plants in the F2 generation and reselecting progenies until genetic purity is achieved. This method maintains detailed records of parent-offspring relationships, known as pedigrees. Pedigree selection differs from mass or pure line selection by using hybridization to generate variability. First described by H.A. Lowe in 1927, it focuses on traits with high heritability, avoiding selection for vigor due to environmental influences. The pedigree method involves crossing parents with desirable traits, growing F1 and F2 plants and selecting superior progeny over several years. This detailed process ensures the development of high-quality crop varieties. Continuing the pedigree method, F6 progenies are evaluated and superior lines are identified through trials. Selected lines undergo adaptation tests, and by the 12th year, they are named, multiplied, and released as new varieties. The pedigree method's merits include maximizing breeder skill, eliminating inferior plants early, and requiring less time than bulk methods. These advantages make it a preferred choice for developing new varieties. However, the pedigree method has demerits. Valuable genotypes may be lost early. There's no natural selection opportunity, and it requires extensive record keeping and handling of many crosses, making it labor-intensive. The bulk method handles generations following hybridization without keeping ancestry records. It allows natural selection to eliminate weak plants, with artificial selection applied to enhance desirable traits. In the bulk method, F2 and subsequent generations are harvested in mass. This method leverages natural selection, differing from pedigree methods by focusing on generation handling post-hybridization. The bulk method procedure involves growing F2 populations, harvesting in bulk, selecting superior plants, and testing progenies for yield. This approach simplifies breeding but requires more time to develop new varieties. Merits of bulk selection include simplicity and cost-effectiveness, while demerits involve longer development times and less efficiency on heritable traits. It offers limited opportunities for breeder skill application. Welcome to Chapter 5, Principles